All right. Right.
Online, when you go to lab 5, you have lab 5, page 1. There's nothing there.
Oh,
When you go on down to the next section, you can get that entire lab. So is that what we're supposed to do?
I'll
or-
find out. I'll take a
Okay.
look. This is all part of that transition from not online to partially online. So I'll take a look.
Yeah, because I printed the other one off. I was going to bring it. And I left it in the office to show you.
So it says there's 2 links on there. Is that what you're saying?
You have lab 5, page 1, nothing shows. Then when you go on down, it'll say, because it's 2 sections, what it looks like. Then it'll say lab 5, page 2.
Take a look now. Got a little time. Yeah, you guys are here for the growing pains. But you know what? Since it's kind of been really beneficial to you guys, it's a lot looser on group. I can't think. I got this week. Due dates. Really loose this semester on due dates. You really shoot for this date, but as long as you get it done before I grade it. All right. Anyway. Interesting to see what you mean now. It says lab 5 pre-lab doesn't have anything. Let's scroll down. Oh yeah. Okay, good question. So glad you asked too, because again, I didn't set this up. I just told them what lab we're doing, when we're doing them. So yeah, just do lab 5 on the next week, because technically nothing's, we're not technically doing the lab next week. Next week we're just meeting. I'm doing the pre-lab lecture, and the week after that we're doing the lab.
So we don't need to do anything Sunday?
Technically, you should be doing the pre-lab, and I'll put out an announcement. So like you said, scroll down where it says photosynthesis part 2, and there actually is a link. Use that link. I'm opening it right now to make sure it looks good. Do that. Watch this video, answer the questions.
Yeah, it's quite long. I looked at it.
Yeah, the lab itself is long. The pre-lab is actually really short though, other than the, even that. So you've got 4 questions on the pre-lab. And then you need to do all the hypotheses, but unlike last lab, there's only 4 hypotheses. But like I was saying earlier, because this is such a growing pain semester, technically, yes, the pre-lab will be due on Sunday, but honestly, between us, you guys are the early students anyway. If you ever watched the video, as long as it's done before the Sunday, before the actual lab. So, not this coming Sunday, but the Sunday.
Okay, then my next question is, when will the exam be released or you?
Well, we still have one more person that needs to take it. So the ball's in their court. The plan is they will be taking it tonight. When I'm looking at these pre-lab questions, it's a good time to remind everybody. Almost always this is the case, but definitely for this lab. All the answers for your questions are going to come from the pre-lab introduction. So make sure you read it and use that to answer your questions. Part of that is just as like proof that you've read pre-lab, you know what I mean? Like, so if you answer something about, first one's about pH, right? If you answered it correctly, but it has nothing to do with what was in the introduction, you get it wrong. Hope that makes sense. Only because, again, this is kind of like proof that you read the introduction. But it's a double whammy. One, I want to make sure you understand it. Two, I want to make sure you read the introduction. Shoot, is it you today? Do you have a pen?
No, it's not due today.
Okay. Look at the grades here. Calculated up.
Thank you.
So when you guys look at your grades online, does it give you a letter grade? No,
No,
okay,
there's a percentage at the bottom.
okay. Oh, does it give you a percentage? That helps. See, when I look, it doesn't do that.
I can go back. Yeah, I scroll down all the way to the bottom and it gives me percentage, but I think the way your class is, we have to work up to a certain percentage, not like we would automatically get an A if we got an A in our lab. You know what I mean?
Yep. So that's another thing we need to fix.
Yeah, so it says I have like a 54%, but obviously I don't have 51%. We just haven't done
Right.
half the stuff that we need to do.
Yeah, for that, 54% would make sense for the whole total since we just finished. Sort of right. No, I don't say that either. Anyway, let me calculate it myself. Okay, it's going down to one. Sure. Takes a little bit off.
Thank you.
All right. In about a minute, but might as well do some attendance. Bryant is not here yet. Curry, not here yet. Darby, here. Fisher, online. Hamilton, here in person. Hellman. No Hellman. Ashley's here in red. What's your credit for that? Crystal's here. Taylor is here. Norris is not here yet. Rollins, not here. Shamblin, here in red. Taylor Pierce, Vargas. Young is online. Sometimes Saunders, who's online. Well, it's 8. We're gonna get a slow start. We're still missing a few people. People who are usually here, so maybe there's traffic. I'm gonna go slow. I guess I'll do announcements. So to remind you, again, I'm gonna be online for office hours. I have to travel this weekend. So that's why, like, as soon as we're done here, I'm hitting the road, but I will be online. So that being said, you'll see the announcement soon. If you need me and you go online to talk to me and I'm not there, that's probably because I just lost service temporarily. So just try back and I'll be back on eventually as soon as I can. What else? I guess that's it, really. Oh yeah. So again, next week's lab is going to be just the pre-lab, right? We're going to meet. I'm going to tell you how we're going to do the photosynthesis respiration lab. I'm going to tell you how to write a lab report. We're not physically going to do any activities next week for lab. It's going to be a lecture. And then the week after that, so I'm physically going to do the labs. So, and I'm glad someone noticed this. If you look at the lab, I wish I had it pulled up. If you look at lab 1 or the lab 5 photosynthesis, it says part 1. When you click it, there's nothing there. And then if you scroll down to the next week and look at part 2 and click it, there is something there. So you can just ignore part 1. Just ignore the fact that it's there. I guess when they made the online shell, they had to have some sort of filler. So you can ignore that. I guess that's it. Any questions? All right. It's 2 minutes past. I guess no one else is coming. Stand corrected. You can see those back there for now. We have just in case I didn't say it out loud for the recording, we got Bryant here and we have Norris here. I think everyone else is accounted for. Yep. So any questions before we get started? So remember we were talking about cells. First we talked about like cells in, well, yeah, cells in general. Then we talked specifically about prokaryotic cells, which is part of what we're concerned in this class, and basically bacteria. And that was the last time we'll probably ever talk about them. And then we started talking about eukaryotic cells, which is what we're talking about now. And then within eukaryotic cells, we started breaking that down between plant and animal cells. Of course, there are other eukaryotes, and we'll talk about that later, like fungi, for example. But we're only focused on plant and animal cells. Like I said, on Wednesday, a lot of the stuff we're covering, I'm only covering it just to give you an introduction to it, and because it's out of the textbook. A lot of this stuff I'm not going to test you on yet, because the real important stuff comes later, in my opinion. So anyway, let's jump into it. We're done talking about plant cells. Let's talk about animal cells. On the outside, they have something called an extracellular matrix. I'm not going to ask you any of this, especially the fact or the details of what it's made of. Let me pull this up so I can scroll through it. Okay, good. But yeah, that's the stuff that basically holds stuff together, right? So obviously you're made up of a bunch of cells. You're not just one cell. You're made up of a bunch of cells. So it's this extracellular matrix that basically holds you together. And also, and this will be slightly important later, but not much. Your cells need to communicate with each other, and this is one of the ways it happens. But I'll put a big X to this to indicate, again, this will not be on the exam, any of the exams. But anyway, since we're talking about it, the first word for attendance is matrix. So make sure if you're online, you send those words in. Within 5 minutes to get your credit. If you guys, if you want to, you can send them in to get your extra credit. Yep. Moving forward, intercellular junctions. Before I talk about it, once again, I'm gonna put a big old X in this, indicating I'm not gonna ask any questions about it. But this is where there's direct contact between cells for communication. So if you want to look into it for independent work, it's not very important, but it is a topic to look into. I just told you that intercellular junctions are direct contact between cells for communication, but then again, I also told you that the extracellular matrix was also used to communicate, right? So if you want to, you can look into the differences. What makes them different? Are they utilized different ways? There's also this plasmodesmata, which is junction between plant cells. And I know we were just talking about animal cells, now we're talking about both. Animal cells include tight and gap junctions and desmones. These are, again, things I'm not going to ask you about, but I'm teaching out of the textbook. Are there any questions about this before I show you a picture of all the stuff I was talking about? All right, here's a picture. So again, you can see how the cells are connected, which in some cases allows things to move back and forth, which again makes sense. These are all things that we need. Again, cellular communication. It's all right there. And again, for the grand scheme of this class, this is really important information. And we are running behind anyway, so we're going to skip through it. So are there any questions about this stuff? All right, so again, if you take notes, I'll write down, don't worry about this stuff. You could completely brain dump all this and understand all the stuff I'm going to teach you later. Here's something that's a little bit more important, the cell membrane. Obviously, it's important enough to where we had a lab about it, right? So this past week's lab was all about diffusion and osmosis. But we had those little things that were supposed to basically mimic the cell membrane. So obviously, the cell membrane is going to be slightly important. The learning objectives for this portion are to understand the fluid mosaic model of membranes. And this one's a little bit more important, to describe the functions of phospholipids, proteins, and carbohydrates in membranes. And even then, a lot of this isn't going to be important enough to take you on. The fluid mosaic model. Big ol' X to this too. I'm not going to ask you. This describes the structure of the plasma membrane as a mosaic fluid of components. So earlier on Wednesday, I said that our cell membranes are phospholipid bilayers, and they're embedded with proteins, and that's basically what we're saying here. But now we're going to get a little bit more specific. But again, I'm not going to test you on this. This is just good things to keep in the back of your mind as we move forward. As far as the fluid mosaic model is concerned, in animal cells, we have a lot of cholesterol in the membranes. That's important for animal cells, basically because it acts as an antifreeze and it regulates the fluidity. So basically, if the membranes need to be a bit more or less fluid, we can kind of control that with the amount of cholesterol in our membranes. Again, and X through that, because I will not be asking this. Like we said earlier, our membranes are mostly a phospholipid bilayer. What I didn't say earlier, and this is not necessarily important as far as the testing is concerned, just keep these ideas in your brain as we're talking about diffusion and osmosis. So the surface of the membrane is hydrophilic, and the inside is hydrophobic. So that should tell you, obviously, hydrophilic things are going to be attracted to the surface. Hydrophobic things are going to be repelled from the surface. And then the opposite is true from the inside. And that's nothing to remember. I guess the point here, the thing you need to keep in the back of your mind is it's going to be complicated, right? When we're trying to get things in and out of the cell membrane, which is what we're talking about when we're talking about diffusion and osmosis and active transport, which I haven't taught you about yet. You just need to keep that in the back of your mind. Long story short, things are complicated. I'm going to put the X through this as well. We can just truck right through this. But are there any questions so far? Okay, so what makes up the second biggest component of your cell membranes? I've already said it earlier, but here it is again. Proteins. Proteins make up the second major component of your cell membranes. And they do a lot of things. I think I mentioned this earlier, but again, you don't necessarily need to know them. They serve as channels or pumps, because again, when we're talking about allowing things in and out of your cells and controlling what comes in and out of your cells, you're going to need channels or pumps to do that. So as far as this conversation is concerned, that's the most important bullet point, because we're going to come back to that. Also, there's enzymes. We haven't talked much about enzymes yet, but I did bring it up when we talked about proteins. far as we're concerned in this class, enzymes are all made of proteins. And basically they catalyze reactions. So there's chemical reactions that need to happen in your body that just otherwise wouldn't happen without enzymes. So that's why enzymes are important. We'll talk about them later in the semester as well. Not specifically just the ones that are embedded in your cell membrane, but enzymes in general. And obviously there's going to be attachments for the cytoskeleton. Because remember, that's, even though you don't necessarily need to remember, that's the stuff inside your cell that gives it its shape and helps it maintain its shape. And it also helps for cell recognition, which is how it makes sense. Because again, this is on the outside of the cell. Cells need to recognize each other and say, oh yeah, that's part of my body, or oh no, that's not part of my body. We need to send something from the immune system to kill it, for example. Anyway, any questions about this stuff? It's getting a big X because it will not be on the exam. Good, we're making time. Third major component. So again, we have the phospholipid bilayer, then we have proteins, and the third one is carbohydrates. Yep, and again, not going to be asking this, but these are always found on the exterior. So if we're looking at the, remember, phospholipid bilayer, right? So if you have a cell membrane, it's a bilayer. I'll try to draw that. And I'll show you a picture of this later, an artist rendition. But so all the other stuff I was talking about, right? The proteins, sometimes the proteins are on the outside, sometimes the proteins are on the inside, sometimes they gap the whole thing. But when it comes to these carbohydrates, they're all on the outside. Again, not that that matters because I'm going to ask you. They're always bound to lipids or proteins, so they're not just sitting there floating on their own. So again, like I said, if you have a protein on the outside here, it might have a carbohydrate attached to it. And this is used for cellular recognition. And again, it's stuff you could brain dump and you'd be fine for the rest of the semester. But if ever one day you go to the doctor and they're trying to say, hey, you should take this new medication for your disease, and you're reading about it, it might have something to do with this. So if you look into it, or if you had to look into it, you're not technically starting from scratch. Phew, got through that. Any questions before we move on to the more important stuff? Passive transport. No questions? All right, passive transport. In a way, I kind of wish we would have done this before the lab. In another way, because you've been exposed to it in the lab, this will be a little bit of a review, a little bit more information added onto it. All right, as you should know from the lab, and you might need to know for the exam, membranes are selectively permeable. You can write it just like that, or you can write down what that means. Probably better. Who cares about what it says verbatim? Membranes allow some things in and out, and don't allow other things in and out, right? That's what that means why we say selectively permeable. Because again, like I said when we started this chapter, cells need to be able to control what comes in and what comes out. There is a link. Shoot, it's not working now. So your lab, on the last one we just did, there was a link that you could click to watch video. How many of you tried that link? Did it work for you? Oh, it did for you. Okay. When I checked it, it didn't work. But I fixed it. So anyway, if it worked, you should check it out. It's a really short video. It was like 12 minutes and says exactly what I'm saying, but a lot more animated. So it's a little bit easier to remember. And maybe even understand. So I highly recommend watching this video. But anyway, yeah. Cells need to control what comes in and out. That's kind of what this picture is like. So whatever these little balls are, they can't pass directly through the phospholipid bilayer. So whatever this is, it just lets them diffuse down. That's what we're seeing in this picture. And of course, I'm going to give you some actual examples of this later. But this is diffusion. This is facilitating diffusion, which you're going to learn about later. This, you need a little extra, right? It's still letting it through, but what you can't see in that picture is it requires energy. But again, these are the things we're all going to talk about. So this slide, again, is basically just an introduction to what we're about to talk about. Membranes are selectively permeable. Cells need to control what comes in and out of them. So I guess that'll be the next word for attendance. I'm going to circle it instead of saying it, so that's the next word. If you're watching the video and you have to take a picture of me when I say it, since I'm not saying it, you can just take a picture of me pointing at it. There you go, or a screenshot, whatever. Any questions about this? All right, another little video you can watch if you download this PowerPoint. This little animation that shows things coming in and out of the cell. This is definitely not as good as the other video that I recommended from the lab, but you can watch it if you want. Same thing with this. That's another animation. It looks cooler because it looks more like more realistic. Watch it if you want. Don't watch it if you don't want. So that's all the information I'm about to give you. All right. As we already know, molecules vibrate and wander randomly, right? In your lab, we call that Brownian motion. For some reason, your textbook doesn't actually define it. But that's where we get diffusion from, right? Because things just bounce around, they diffuse from an area of high concentration to an area of low concentration. And what I mentioned it a little bit, but I didn't emphasize it last week or in the lab. This happens until equilibrium is reached. So you need to know that. This is an important bullet point. Diffusion is the movement of molecules from an area of high concentration to low concentration. And I'm even going to write the word dynamic, even though the only reason I'm doing this is because I think on the study guide, that's how it's written. Dynamic equilibrium is reached. Which I'll probably add in the study guide. If it does say that, just get rid of that word. Basically, again, like I said in lab, the example was, all right, if I light a candle here, a scented candle, you guys won't smell it yet, right? But if it's going to be really heavily scented to me, but eventually it's going to diffuse throughout the room to where everybody's getting the same amount of scented candle, right? That's when equilibrium is reached. Another important thing is that it does not require an input of energy. Right? So again, if I lit a candle, I mean, it would be quicker if I wafted it, but that is an input of energy. I could just light the candle and let it go. So you guys who did experiment 1, you put that drop of dye on the plates, and it just naturally did its thing, right? It naturally diffused. You did not need to put energy into it. Now, granted, when you did put energy into it for the one that was on the hot plate, it went quicker. But it does not require an input of energy. This just naturally happens. Yeah, so remember that for the exam. Sorry, let me just go back to this too. This picture is supposed to demonstrate that, right? Molecules vibrate and wander randomly. And they do, and you don't need to know this, but this is just a great example of a water molecule, for example. Try to picture, well, that's a water molecule. Try to picture this thing going like this, right? It's not sitting still, it is right here, but imagine it's stretching and contracting like this, right? Imagine this one doing the same thing, but like this instead of at the same time. Imagine this one kind of going up and down, like flopping its wings. This one's rotating this way. This one's rotating this way. This one's rotating that way. So there's all these different things these molecules do. And imagine again, if those molecules are sitting next to each other and they hit each other, then they're going to go flying off. Does that make sense? So again, this is like a visualization of Brownian motion. It's not that I'm going to ask you about all those different types of movement, but are there any questions? All right, a visualization of diffusion. Here we go. Here we have these molecules of dye. We have this membrane that's allowing this dye to go back and forth. So what you can't see, and your book tried to make it look like X's in another textbook, but they're blurry for a reason because they're moving around, they're bouncing around each other. And imagine those are just a bunch of pool balls, just kept bouncing, right? And eventually they start coming over. And when we use the word dynamic equilibrium, like in your study guide, what that means is, well, first of all, let's just talk about equilibrium. There's equal amounts on this side as that side. Does that make sense? That's equilibrium. Dynamic equilibrium just means it's not like we get 1, 2, 3, 4, 5, 6, 7, 8. It's not like we get 8 balls on each side and then we're done. What's actually happening is, even though equilibrium is reached, they're still bouncing around. And some of them are still going on that side, and some of them are still coming to this side. But equilibrium is reached, and the reason it's dynamic is because, yes, the molecules are still moving around, but equilibrium is reached because the concentration is equal everywhere. So any questions about this slide? So again, remember, area of high concentration to an area of low concentration. That is net diffusion until equilibrium is reached. And then your other textbook also showed this. It's kind of the same thing, except for 2 different color dyes. So here you got the purple ones that are doing a net diffusion to the right. And here we have the blue, red balls for the net diffusion to the left. And again, still dynamic equilibrium is reached. Anyway, any questions about that? All right, another thing you should know for the exam, and you're already familiar with from lab, is this: the factors that influence the rate of diffusion. Concentration gradient. So what did we determine? Did we determine starch did or did not diffuse out? You guys remember? Or sucrose? Did sucrose diffuse out? You guys remember that one? Salt? Salt definitely diffused out. All right, we'll use that as an example. So imagine you have a beaker, water, and you've got a little cell in there, and it has a salt solution. And the salt solution is, I don't know, 80% salt solution. And the water is 0% salt solution. It's just pure water, right? And then you have another beaker. Same thing, same setup. It's got some water with 0% salt in it. But this little cell that you made only has, I don't know, 10% salt solution. If you had to guess, which one's going to be diffusing quicker? Now, eventually, I'll tell you what's going to happen. You're going to end up with an equilibrium, right? So it's going to be equal. But which one's going to be diffusing faster? A or B? If you had to guess. And I haven't told you this, so don't feel bad if you don't know it. This is a guessing game. What do you think? Obviously, this is going to diffuse out. Salt's going to diffuse out for both of them. Which one do you think is going to happen quicker? The rate of diffusion. What do you think is going to happen quicker? I would say this one. Going back to what we're talking about right now, that first bullet point. The concentration gradient is greater. We're going from 80% to 0%, right? So it's going to be coming out quick. Here, it's not going to be coming out as quick. So 10 to 0%, not as big of a difference. Does that make sense? Almost imagine like if you had, this is kind of taking it a little bit too far. If you had a dam, right, with some water behind it, that's where the water comes out, and then the river, right? Water's going to be coming out really, really fast in this dam. And if you have the same dam set up with less water, water's going to be coming out a little bit slower. That makes sense? Concentration gradient, right? There's a lot more water trying to diffuse from an area of high concentration, low concentration here. And here, there's a lot less water trying to diffuse from an area of high concentration, low concentration. Make sense? All right. So that's the concentration gradient. That is important. Mass. That's not much to say that it's hard to picture, but again, it's just molecular mass, right? The bigger the molecule is, the slower it is. It just takes more energy to move something larger, right? Or you can almost visualize it like this. So they were using little plates that had agar in it, right? A gel. So imagine like something trying to get through gel. The smaller it is, the easier it is to get through gel. Or a bullet, right? And forgetting its propulsion. Something larger, a larger bullet would be harder to get through air than a smaller one. So any questions about concentration gradient or mass? Temperature. We also did that in lab too. And that just makes sense too, because again, temperature, we're just talking about molecular motion. So again, when I say the temperature of the water is rising, what I'm saying at the molecular level is those water molecules are moving around faster. So obviously if things are moving around faster, they're going to move around faster. They're going to go from an area of high concentration to low concentration faster. They're going to diffuse faster. And then again, solvent density, and we don't talk too much about that one. I won't get too much into it because I'm not going to complicate it. I won't ask you this. Solvent density is almost in a way the opposite of mass. So when we say mass, we're considering the mass of the stuff that's going to be dissolved, right? Stuff that's going to diffuse. Here, solvent density, we're talking about the stuff that it's diffusing through. So for example, again, if you're in group 1, use those gel plates, right? They have like a little jello looking stuff in it. Now, if you were to do the same thing with a plate that had water in it, right? Imagine you put a little drop of dye in that water, it's going to diffuse quickly, right? Because it's just water. It's not as dense as the gel that they were using. Does that make sense? All right. Does anybody have any questions about those 4 things? All right. And again, this is largely a review. If you download the PowerPoint, you can watch this little animation of diffusion in action. All right. So sometimes substances do not cross spontaneously. right? Even though they want to diffuse from an area of high concentration to low concentration, sometimes they can't. And this is where we need proteins. Certain proteins allow this to happen. That's what you can see right here. And this is called facilitated diffusion. You need to know that for the exam. So under the term passive transport, which can be abbreviated, under that term, we also have something called diffusion. And then one of the types of diffusion is passive transport. Excuse me, I said passive, going the wrong way. Facilitated diffusion, excuse me. So yes, facilitated diffusion is a type of diffusion versus a type of passive transport. But again, it's just diffusion except for whatever reason, it cannot cross the membrane. So like I said earlier, remember the outside is hydrophilic, the inside is hydrophobic. I said that's going to come back later. And this is a good example of that, right? So for whatever reason, chemically, there's something about it that won't let it go through, right? These little purple balls, they can diffuse right through. Something about the chemical of these little triangles makes it to where they cannot go through the membrane. So they need that. So for example, imagine if the doors of this room were open and we fit everybody on campus in this room right now and turn on the heat, right? People are naturally going to want to go from this area of high concentration out to the hallways, right? There's too many people. It's going to smell bad. It's going to be hot. They're going to naturally want to do that. Now, if we close the doors, they're still going to want to do that, and the air is still going to do that. The air will still slide down through the crack of the doors, right? It'll still go from an area of high concentration to low concentration. But humans cannot get through doors, right? We have to open them. So you can consider the door basically the protein, right? So it allows things in and out. So again, facilitated diffusion is a type of diffusion where something cannot make it through the membrane, so it needs help. Doesn't need energy, it just needs like a passage way. So that's another thing to emphasize too. Because we're still talking about diffusion, it does not require an input of energy. And I just put that there as emphasis, because like I said earlier, if it's a type of diffusion, we already know diffusion does not require an input of energy. So obviously, facilitated diffusion won't either, because it's a subcategory of diffusion. Anyway, any questions about facilitated diffusion? All right, there will be questions about that on the exam. Facilitated diffusion. So we're still going from an area of high concentration to low concentration, but for whatever reason, the solute, the thing that's being diffused, can't make it. Can't go through the membrane. So it basically needs to pass through it. All right, no questions about that. Let's get into the one we didn't talk too much about. We brushed up on it. Let's talk about it in a little bit more detail now. Osmosis. That is the diffusion of water across the selectively permeable membrane. I think everyone got that right. I'll do that. Diffusion of water across a selectively permeable membrane. If you just leave it at that, that's not very complicated. Because basically it's everything we've been talking about, except now we're all of a sudden getting specific. So I'm not talking about anything going across the membrane. I'm talking about water only. But before we can have this discussion, and before we make it a little bit more complicated, there's some words you need to know. I told you before, there's actually, I think, questions on it on at least one of the study guides that you've already had. Maybe even on the first exam. Anyway, these words you need to know, at least so you understand what I'm saying, and probably for the exam too. A solute is a substance that is dissolved in a solvent, and the resultant mixture is called a solution. So if you mix tea and like tea leaves with water to make tea, the tea leaves would be the solute, the water would be the solvent, and your tea, would be the solution. You mix sugar and water to make simple syrup. The sugar would be the solute, the water would be the solvent, and the simple syrup would be the solution. Make sure you understand those, because we're going to get a little bit more complicated with osmosis, and I'm going to be using those words. So does anybody have any questions about what those words mean? All right. If you download the PowerPoint, you can watch this little animation that shows osmosis. Here we go. Forget for a second that we are talking about osmosis. Forget that. Let's pretend you can just ignore all the words on there. Just look at the picture and pretend like we're talking about diffusion. All right, so forget all the words. We're talking about diffusion, not osmosis, just diffusion. Knowing what we know about diffusion, what do you think If we're only talking about diffusion, which way should these balls be going? Should they be going to the left? Should they be going to the right? Should they be staying where they are? Where do you think those balls would go when we're talking about diffusion? Right.
Or no, look, it's the left.
To the left, correct, right? Because here we have a high concentration and here we have a low concentration. I know it might be hard for you guys to see. So this is an area of high concentration. This is an area of low concentration. We know, maybe where I hear the arrows might have thrown you off too. We know that those balls want to diffuse to the left, from the right to the left, right? They want to go from an area of high concentration to low concentration. Now let's talk about osmosis. Here we have this semi-permanent low membrane, and it will not let those balls move over. So how can we have an equal out, right? How can we have an equal amount of solute? Let's say, and I'm just making this number up, so we can visualize it, but let's give it a number. Let's say this is those green things. Let's say it's sugar. Why not? And let's say that's, I don't know, 60% sugar, obviously 40% water. And then let's say this is 40% sugar. How can we equal those things out? How can we make it to where that's 50 and that's 50? What do we do if we can't move the green balls over? Any guesses? The water will move instead. So the water is going to move from left to right. And now, I mean, we've got a lot more water over there, but now we're at equal solution. I'm just throwing those numbers out, right? So 50%. And again, just throwing those numbers out there. The point is, it's equal solution, right? You can see now it's equal concentration. That makes sense, everybody? This is why osmosis happens. This is why the water moves from an area of high water concentration to an area of low water concentration. But here's where it gets more confusing. When we're talking about diffusion, osmosis, we change the words up. So in diffusion, osmosis, can't read that, so let me put it here. What's happening in osmosis is it's going from an area of lower solute concentration to an area of higher solute concentration, right? So I know that sounds confusing because now we're going from an area of low to high. And that doesn't make sense, right? Because we're talking about diffusion. When we're talking about osmosis, we're not referring to the water, we're referring to the solute. Which brings me to this. You need to know these words too. When comparing something that has a lower concentration, we call that hypotonic. If it has a higher concentration, it's called hypertonic. When it's equal, it's called isotonic. You need to know those words. And there is a question on the study guide. There will be a question on the hand that's similar. It basically says, I don't know, the numbers are going to be different, but here, let's look at this one. That's 10%. Change it up a little bit. Make it a little bit more complicated. Let's call that 20% solution, whatever it is, sugar, I think we said, and that's 10%. How would you describe it? So you could say that this cell is hypertonic to the beaker. Does that make sense? Because it's a higher concentration in the cell than the beaker. Likewise, you could compare it the other way and say the beaker is hypotonic to the cell. That makes sense. It's a lot like pH. We're describing the same thing, and you can use different words to describe it. And on the exam, that's how it's going to be. You're going to be describing it in different ways. And it's all relative. For example, if we were to all stand up right now, you guys would probably describe me as tall. Or at least taller than you guys, right? He's tall. But then if my younger brother were to come in, he's taller than I am. So all of a sudden, he's the tall one. Right? Does that make sense? Because it's all relative. So again, using this as an example, that one's 20%, that one's 10%. So yes, that one's hypertonic. But then if you were to switch it out, put one in here that's 5%, now all of a sudden it's hypotonic and the beaker's hypertonic, right? We didn't even change what's in the beaker. We just changed the cell and now all of a sudden it's a different description. It's all relative. These are only terms we can use to describe different things, comparing them to each other. So does anybody have any questions? It's very important that you understand hypotonic versus hypertonic versus isotonic. I think one of the easy ways to remember that is like when a kid's hyper, what does that mean? Generally speaking, when you say, oh, that kid's hyper, how are you describing him? Full of energy, right? That's one way you could put it, right? He has a lot of energy. Too much almost, right? Kids bouncing off the walls. Too much, right? More than usual. That's hyper. And then if you know other like medical terms like hypoglycemic, that might help you, like low blood sugar. But I think the easy way to remember that is kids. When a kid is hyper, they have more energy than usual. And again, that's relative too, right? You're usually comparing them to themselves. Anyway, any questions about that? All right, so this next slide is everything I just said, but in writing. And there's a word for everything I just described, and I'm not going to ask you this word. It's called tonicity. So all the stuff I've been describing and teaching you is called tonicity. I'm putting next to it only because I'm not going to ask you what tonicity is, and I'm not going to use that word again. But you're booked in, so there it is. So yes, again, same with osmomolarity. Basically saying the same thing. So tonicity describes the amount of solute in the solution. Osmomolarity is kind of like the same thing as tonicity, except it's specifically in the case of osmosis. Those are words you don't need to understand about. But you need to understand, again, hypertonic is a higher concentration, hypotonic is a lower, isotonic is equal. And iso should be easy to remember because isomer, which is a word you needed to know, right? So the other iso word that we did. I don't have time to think about it right now. But anyway, any questions about this slide? All right, this brings us to why this is important for biology. Because to survive a hypotonic or a hypertonic environment, your cells have to balance the uptake of lost water. In other words, you cannot be a free-for-all. Just like anything else, your cells have to regulate what comes in and out of it, right? And that's where we get osmoregulation. That might be a word that you should know, osmoregulation. Kind of in the word though, or it's in the name. Osmo, short for osmosis, regulation. So it's using osmosis to regulate the amount of water, control the water balance. So right now, by the way, we were talking about animal cells because it's different from plant and animal cells, and you'll need to know the difference for the exam. But for animal cells, we prefer an isotonic, generally speaking. They prefer an isotonic environment. And I'll show you a picture. When I show you a picture, it'll make a lot more sense. And it's going to show you all these things I'm about to describe. Because if it's in a hypertonic environment, it's going to dehydrate. Because, again, picture it, and I guess I'll just try to draw it, even though you're about to see a better picture. If this animal cell has, 10% of whatever solute, salt, sugar, whatever it is, and then it's in some environment that's, 80%, doesn't matter the exact numbers. The point is, it's in a hypertonic environment. The environment is hypertonic to the cell. So what's going to happen? Well, the water is going to want to leave the cell to try to even things out, right? So as the water leaves, this number goes down, this number goes down, this number goes up. Again, it's just trying to reach that balance. So that's what would happen in a hypertonic environment. The opposite would happen in a hypotonic. And again, these numbers don't exactly matter. The fact is one's bigger than the other. In a hypotonic environment, let's see, that's 80% on the inside of the cell. And maybe in the environment, it's 10%. So again, the water is going to want to move to balance things out. Like it doesn't want 10%, 80%. It wants 50-50 as close as it can. So water in this case is going to leave the environment and come into the cell. But as you can imagine, just like a water balloon, it can only hold so much water before it explodes. So I'm going to show you a better picture of all this stuff I just drew. Before I do you guys have any questions about the words that are on the slide? Actually, no, I don't have a picture of that yet. I will, but on the next slide, we're going to talk about plant cells. Yeah, so any questions about this slide? All right, plant cells are different. Actually, before I say that, this is a good independent work topic. So everything I just told you is generally true for animals. But then think about some other animals a little bit more complicated. For example, what happens if you put a freshwater fish in salt water? Or what happens if you put a freshwater or saltwater fish in freshwater, right? It'll kill them. So for independent work, generally, there are some that can handle it. Like salmon, they don't care. Bull sharks don't care for the most part. But anyway, you can look that up. What is it about fish, right? What happens when you put a saltwater fish in freshwater fish? Or vice versa. Things to think about. Or if there was a great flood, the whole world was inundated with salt water. And what happened to all the freshwater fish? They were inundated with saltwater, things like that. But let's talk about plant cells. So what I'm still saying is true, right? In that when any cell is in a hypertonic environment, the water is going to leave the cell, right? No matter what, generally speaking, whether it's plant or animal cell. Same thing with if it's in a hypotonic environment, water's still going to come in. But remember, one of the differences between plant cells and animal cells, I told you this would be important later, here it is, they have cell walls. So because of this, not only can they take water coming in, but they prefer it. That's what keeps the cell or the plant rigid, so to speak, turgid. It needs the water constantly coming in to help expand it. Sort of like those stupid wiggly arm things. You ever seen them like outside of a used car sale, a used car place, right? It needs that constant input of air so it can stay at least semi-turgid, right? Actually, if you put enough air in it, it'll probably be just like that, right? But if you don't have enough, then it will just flop down, right? Same with this. With plant cells, they need that water coming in. Therefore, they prefer a hypotonic environment. So the water is trying to go in. Isotonic is not as good because the water coming in and out is equal. It's not necessarily bad for it, but not good. But then hypertonic is obviously going to be really bad for the same reason it's bad for us, because it gets dehydrated. But in this case, not only does it get dehydrated, but then the cell membrane also detaches from the cell wall, which is really bad. Any questions about that before I show you just some visualizations of this? All right, here you go. I'm sure you guys have seen this before. I wish I could find my video. I had a plant like this, and I left. My wife didn't water it for a while, and it looked like this. But then I watered it, and I took one of those videos where it's sped up. So like 30 seconds I went from this to this. It's a neat looking video. I wish I could find it. But anyway, that's what happens, right? Once they become dehydrated, they shrivel. But once they have enough water, they expand, right? The water coming in, pushing those cell walls out. That's what happens. So any questions about that? To sum it all up, what you need to know for the exam is this. Mostly you need to know animal cells prefer isotonic. Plant cells prefer hypotonic. Put an A here for animal, P here for plant. So all the stuff, I gave you all the explanations of why and all that, but when it all boils down to the exam, you need to know isotonic is good for animals, hypotonic is good for plants. And yes, I explained why. Hopefully that'll help you remember. Right? Too much water in an animal cell and it explodes. Not enough water in an animal cell and it's dehydrated, right? Plant cells want a lot of water to push it out. Anyway, any questions about that? We're making good time. We might be able to finish this chapter and then be caught up. Active transport. So everything we've been talking about has been passive transport, right? All this osmosis, because that's just a type of diffusion. All the passive, all the facilitated diffusion, all the diffusion, that's all been passive transport, right? It's just things moving across membranes without any kind of input of energy. Now we're talking about active transport. The learning objectives of this are that you'll be able to understand how electrochemical gradients affect ions. And I'm going to go ahead and tell you this, that's directly from your textbook, but I'm more interested. Nah, keep it like that. Explain it later. You should be able to describe endocytosis, including phagocytosis, pinocytosis, and receptor-mediated endocytosis, and understand the processes of exocytosis. So here we go. Let's jump into it. Active transport. What you need to know about that is it requires energy. So there'll be a little bit of comparing and contrasting in the exam, like the differences between active transport and passive transport. And that part should be easy. Someone's going to miss it. And I'm going to have a little tear in my eye because that's the easiest part. It's in the name. Active transport requires energy. Passive transport does not require an input of energy. That should all make sense. That should be, I won't say common sense, but intuitive. Active transport also requires a transport protein. Might or might not ask you that. So remember, with diffusion, sometimes you can come directly across the membrane. Sometimes it also needed a transport protein, in which case it was facilitated diffusion. But in active transport, it always requires the transport protein. And with the other stuff, and I wish I wouldn't have had it on the board, I wish I had time to ask you. Hopefully you would have gotten this. But remember, active transport is essentially the opposite of passive. So here we're going against the concentration gradient, which is why it requires energy. You're going from an area of an area of low concentration to an area of high concentration. Then again, if you watch that video on last week's lab, he does a great job of explaining why, right? So your cells need stuff, right? So if your cell, I don't know, your cell needs potassium, there's not a lot of potassium in its environment, but there is a good amount in your cells. Potassium doesn't want to come into your cells because you already have it, but you still need more, right? So you need a way to pump it in there. Oh yeah, which is basically this little point here. Active transport maintains concentration of small solutes that differ from environmental concentrations. Again, that's what I'm saying is you're going to need these solutes, even if there's a small amount of them in the environment and a large amount of them already in your cells. Anyway, we're going to talk more about active transport, but for now, are there any questions about that side? All right, the next word for attendance, I'm going to circle it again. That's going to be a really important word. You're going to be tired of this word after the next two chapters. Actually, if I had to boil this chapter down into one sentence, I would say this: cells require ATP. Of course, your book doesn't do a good job of saying that, but yeah, cells require ATP. And now this is the first time we've actually mentioned it. There's your word. So if you're watching the video, you take a picture of me pointing at the word. Anyway, let's talk about it. The electrochemical gradient. I'm more interested in the chemical gradient in a sense, but like your old textbook, the one I used to use, it was a little bit easier. It didn't dive into the differences between the electrical and the chemical, but we'll go ahead and talk about it. The interior of the cell is technically negative relative to the exterior, generally speaking. So long story short, there are reasons other than the concentration gradient that necessitate active transport. You don't need to necessarily know this. I'm not going to ask you anything about this. But the point being is, right, so even if you're trying to get something in your cell, I don't know, whatever this thing is, there's your cell, and you've got 10% in your cell, but it's also, I don't know, 10%, so it's isotonic, right? So when we talk about concentration gradient, there is no need for, right? There would be no need for diffusion. It's already equaled up. And all this is saying is even though it might be equal, it's not all about concentration gradient. Sometimes it's also about the electrical story, right? This one's negative, this is positive. So again, I don't think you need to know this. I'm only bringing it up because it's in your textbook. And when you read your textbook, which I'm sure you are, you need to see that. Heard a noise. Let me check on that. Anybody online have questions? Heard a noise. Must be good. All right. Anyway, let me get back to this. Sorry about that. So anyway, any questions about that? Again, not important. Just to see your book, so I included it. If you download the video, you can watch this little animation about active transport. Everything I'm about to tell you. Here's a picture of it. A really basic picture. So here you can see whatever this solute is, your cell needs more of it. Even though it already has a bunch. It has more on the inside of the cell than it does on the outside of the cell. But whatever it is, your cell still needs it. So if you're going to go from an area of low concentration to an area of high concentration, you're going to need energy. And in this case, it's usually ATP. So we expend a little energy, that protein pumps it in, and there you go. That is active transport. That's basically all you need to know about active transport. I will say this, in the next two chapters, this will be important in a way because there's going to be some situations where we're pushing things against the concentration gradient, and it's going to require energy to do that. So just that, keep that in the back of your mind. We'll talk about it in here. So again, for the exam, this portion of the exam, you need to know that active transport requires energy, an input of energy, and it goes against the concentration gradient. So essentially, it's the opposite of passive transport. Now let's talk about the easy stuff. And there are some questions about this on the study guide. I might or might not ask you about this on the exam. It's not that important because after this chapter, we'll never talk about it again. But endocytosis. This is technically another type of active transport. Basically, it's bringing in a bunch of stuff at once. Endocytosis is basically bringing in a bunch of stuff at once. So you can see here, this is a great picture of it from the other textbook. Basically, this stuff, like your cell membrane kind of like envelops it on itself and brings it in. That's all it is. Now, it itself is broken out into two different types, phagocytosis and pinocytosis. The first one is basically cellular eating. So that's when your cells are engulfing food sometimes, but basically solid stuff. Pinocytosis is when it's bringing in liquid stuff. It's that simple. Who knows what Pinot Noir is or Pinot Grigio? You guys never heard that? You do? What is it?
Is it little champagne or?
It's a wine, right? And what do you do with wine? You drink it. So if you can remember Pinot Noir or Pinot Grigio, and remember that it's a wine and you drink it, then you can remember pinocytosis is cellular drinking, and therefore a process of elimination. The other one is cellular eating. We won't be able to finish this. It's 8:50. All right, the last word for attendance is pina colada, which is also a drink. It sounds a little bit like pina. So yeah, I guess that's it. I will see you on Monday. I'll be online for office hours. And on Monday, we will finish up this chapter. Starting there.
Yeah.